It is my privilege to welcome our first speaker, Dr. Courtney Noah, Vice President of Scientific Affairs at BioIVT. Dr. Noah has over a decade of experience in the biotech and life sciences industry and has been at the forefront of advancing BioIVT's scientific outreach, such as, for instance, today's symposium, which was her idea. She also plays critical roles across multiple different areas of the company. Most importantly, Courtney is driving BioIVT's innovation in biomarkers, liquid biopsy, and neuroscience research. Prior to leading the scientific affairs team, Dr. Noah was Senior VP of Marketing, where she was instrumental in building brand awareness and successfully integrated several acquired companies. She holds a bachelor's in food science from Cornell University and a PhD in cellular and molecular biology from Stony Brook University. And before joining BioIVT, she held key positions as a senior product manager and marketing manager at Enzo Life Sciences, as well as Paul Corporation. So today, Dr. Noah will give the opening presentation of our biomarker and diagnostic sessions. And I'm very excited to hear her insights on translating diversity and longitudinal data into discovery. So please join me in welcoming Dr. Noah.
Thank you, Tina. All right, so I'm very happy to be here. So Tina kind of stole some of I was going to give an intro, but I don't think I need to cover that. So I think maybe a little bit in the context of neuroscience to start off. As you can tell a little bit from my background, I am not a neuroscientist by training, actually did more work cellular biology and microbiology, but over the course of my career in life sciences have become a great generalist. But across all of that work, one of the things that has really kind of inspired me to give this talk today, and I think it's something that I'm very passionate and kind of pursuing as a theme here at BioIVT, is how can we improve the aspect of diversity in the research that we do. And it's really come to the forefront, I think, as we think about precision medicine and how much innovation is being done there, the importance of making sure that we have cohorts that we're looking at that are really representative of the disease. And so that's what I'm going to focus on a bit more here in my talk, giving some examples of what we see in the neurospace and honestly some of the gaps and challenges that exist that I think from a biospecimen provider perspective, hopefully we give some potential solutions in addressing those gaps and challenges. So as Tina started out, just saying the impact of neurodegenerative disease is very significant. It affects millions globally. And so today I'm going to speak about the significant disparities that exist specifically around race and ethnicity and sex and gender, and how those have been shown to impact disease prevalence, presentation, progression, and response to treatment. And when we look at studies across drug and diagnostic development historically, there's really significant implications that we're seeing and how lack of diversity in those studies are impacting the generalizability of findings, and really also impacting the confidence of potential interventions that could benefit those populations that are most at risk. So how do we recognize those gaps? How do we develop a path forward? I hope that you'll take away from this talk some considerations on how to design a more inclusive research study and strategy that will ultimately improve the outcomes for patients with neurodegenerative diseases. So not to spend too much time on this, but just to give you a preview into what I'll specifically be covering, I believe there's an opportunity for us to incorporate both diverse and longitudinal samples in neurological research and improve, like I mentioned, that experimental design, really driving to deeper insights that help us to tackle that fundamental goal of precision medicine. So to start, I like to make things personal, just kind of giving an example of a potential real-life scenario here. So this is Eleanor's story. Let's imagine she's a vibrant 60-year-old African-American woman, and she started to experience some memory lapses. So her first indication is, okay, I'm going to go to the doctor and have this checked out. Well, the doctor proposes let's do some testing for Alzheimer's disease, and in that, maybe they're looking at tau protein levels in CSF or blood, maybe, or maybe looking at phosphorylated tau. But in that, Eleanor faces an immediate challenge right there. So if we look at studies, they actually show that African American individuals have lower levels of tau and phosphorylated tau biomarkers compared to white individuals. So this means that when we look at cut points for these tests, they're largely developed actually from white cohorts. And so with that, and Eleanor being African American, that can lead to misinterpretation of her results, can lead to ambiguity or potentially a misdiagnosis. Now for Eleanor, she actually has a secondary challenge there as well. She's a woman, and so as part of this, she encounters another layer of bias. Her strong verbal memory skills, a known female advantage in these types of tests, can make standard cognitive tests too easy for her in the early stages of her disease. So again, what this could lead to is a potential delay in diagnosis, and as we know, with newer therapeutics that are coming to market, they're most impactful at the early stages of disease. So Eleanor's story really powerfully illustrates that when research lacks diversity in race and sex, our diagnostic tools and treatments can fall short for many within the population. And this underscores the need for more inclusive research frameworks as well as biomarker studies that really accurately reflect the population and prevalence of disease, paving the way for that health equity. So let's learn a bit more about where we actually struggle in terms of proper patient representation. And I thought I'd walk us through just specifically what we know today. So if we look at the influence of gender, and I'm going to focus in more on AD and PD. There's a variety of different neuro diseases that we'll be talking about today, but just in the context of this 30-minute talk. So if we look at Alzheimer's disease, it disproportionately affects women. Parkinson's disease, in the contrast, is diagnosed more frequently in men. But despite these kind of opposing prevalence patterns, both diseases certainly exhibit significant gender-based differences in their clinical presentation, their progression, and underlying biological mechanisms. So if we look from a racial perspective, who else is disproportionately affected? Black Americans experience an increased risk of dementia. They have a 64% higher rate of progression to AD and related dementias compared to non-Hispanic whites. But despite the higher rates of developing Alzheimer's, it's kind of interesting that Blacks and Hispanic adults are actually less likely than white adults to be diagnosed with the condition. And so finally, the challenge of underrepresentation. We know it exists. Ethno-racial groups have been historically underrepresented in clinical trials, and I found this one really kind of startling statistic that in one study looking, again, at representation, there's only 1.6% African American participants in a phase 3 trial for Alzheimer's. So that's definitely a problem and a gap that we need to fill, and it really underscores, again, the need for more careful consideration of the influence of diversity in this research space. So in addition, as we think about the context of gender and race, it's also kind of wrapped up within the social and lifestyle risk factors. So these are important determinants of risk and progression for both AD and PD, and they point to the fact that really beyond just the biology, there's these profound other factors that influence the development of disease. So I wanted to call out a few interesting examples that I came across as it pertains to Parkinson's, and honestly things I wasn't aware of. So one is around pesticide exposure, and it's kind of linked to occupation and how that can influence development of disease. So men are actually more likely to be exposed through occupations such as farming to spray pesticides, and that's been linked to an association between pesticide exposure and PD development reported in men. From a female perspective, there was a Canadian study that looked at women with PD that had a stronger preference for a Mediterranean diet compared to men. And they actually found that the dietary patterns associated with later PD onset they saw that in both genders, but actually there was a larger effect observed in women who were following that Mediterranean diet. This stat was kind of interesting to me because I think we hear a lot about how we should reduce alcohol and smoking, but in fact, from a lifestyle risk factor, there are several lines of evidence that indicate that alcohol intake and smoking can actually reduce PD risk in men specifically to a greater magnitude than women. And so these lifestyle factors such as physical activity, diet, smoking habits, they're all important in that they're modifiable. They can be strongly influenced by gender, race, or ethnicity, and they can affect both the onset and progression of disease. So I wanted to pause here just before going through this slide just to say, how did I become kind of interested in this specifically in the neurospace? And a few months ago, I was putting together a poster. I was looking at biomarker differences within our neurospecimen cohorts that we have. And I was interested in kind of doing an assessment looking at divergence by race, gender, age. And just to get myself up to speed and look at the background there, it was kind of surprising to me that there were so few studies in this space that had been done. And then if there was information, in some cases it was conflicting. You know, you would read one study that says maybe this is a difference, and another maybe it's not. And so it was really inconclusive. And so not only is this a problem that we're hearing it talked about, I think, in the broader context of how we design clinical trials and the types of research we need to do overall in the biotech and pharma space, but I think there's a very strong prevalence in the neurology space. And so as all of the technological advances are happening, and I think there's a lot of exciting research that's being done from a precision medicine perspective, I think it's important that we're all kind of doing that checkpoint to say, are we including the right cohorts in the studies that we're designing to make sure that it's really going to be benefiting the populations that are at risk. And so if we look, just as an example of, like I was saying, some of the lack of information that's out there, this first example is Northwestern Medicine study that analyzed over 20,000 US clinical trials between 2000 and 2020. And they found that neurology trials specifically, but also oncology, immunology, and nephrology, had the lowest female representation relative to disease burden. And so they found that women experienced a disproportionately high burden of disease, yet their representation in clinical trials was lower than expected. And then similarly concerning, there is a recent literature review analyzing thousands of Parkinson's disease risks published between 2000 and 2024, and they revealed that less than 5% of these investigations actually looked at or incorporated race and ethnicity into their analysis, and that the majority of studies, nearly 60%, did not account for racial or ethnic factors in any form at all. So with that, like the title says, we do have a long road ahead, but I don't want to leave it on a down note because I think we know the history, we know the gaps, but I think there is a way forward and actions that we can take to improve. So some thoughts for consideration here, and actually some steps that from a BioIVT perspective, we definitely take into account as we're thinking about our clinical site development and how we engage with our patients that are donating samples. So actively work to include those underrepresented populations in research cohorts. So as Tina was mentioning, we're here to kind of have those discussions with you. What does the epidemiology look like in the disease area that you're studying? How does that match up with samples that we have in our inventory? That's something we're here to support and help, and I think it's an important message that we want to convey, especially in the neurospace. From a clinical trial perspective, I think it's critical that recruiting participants not only across different racial groups, ethnic groups, and genders, but also, as I was talking about, from those lifestyle factors and influences, making sure that there's that spectrum of socioeconomic status, education, which is something we actually capture that information on our samples specifically, health conditions, and making sure that really that we're applying all that information to improve the generalizability of the findings. We want to facilitate participation. So kind of one approach to that is how can we overcome some of the barriers in just general participation in sample donation or clinical trials. And I think a key call out here is for considerations around less invasive collection methods. So while we do CSF collections, we also do a number of match paired collections as well that are more blood-based. And so I think as you think about how to facilitate that participation, thinking about things that enable more accessible testing, perhaps less costly, less intimidating, can really bring down the burden of those procedures and increase participation from some of those marginalized populations. It's important that we're building the trust and engaging communities. There's many organizations out there. There's many patient advocacy groups. We also, engage with them as well. And I think the more information we can provide around the research that's being done, how that's benefiting the patients is another critical approach to kind of developing that path and improving the process of better engagement. Another call out is around data collection, enhanced data collection. So the critical nature of making sure that we have comprehensive data, not just the demographic, but including relevant information around lifestyle factors, occupation, even hormonal factors, which has been seen to influence disease progression. And so it's important we also get some of those nuanced information, things like stress, access to healthcare. These are definitely factors that can influence health, significantly influence health disparities and can also mediate some of the differences that we see across biomarkers. And then in the context of what we're seeing, I think in the digital age with AI and machine learning, making sure that the data is standardized and that, again, it's representative. So there's established guidelines out there in terms of how to report proper ethnicity, race, gender information. That's actually through the FDA, which is something that we adhere to. And again, making sure that it's well curated, documented, and essentially analysis ready to feed into these new digital tools that are coming to market. And then finally, informing the research design. So I mentioned there's conflicting data that's out there, but I think making sure that we're looking at biomarker cutoffs across those diverse populations to overcome some of the problematic issues that we're encountering today. So a better understanding of the complex biological and social determinants is really crucial for improving PD and AD care. So I'm going to dig in a little bit more around the biospecimens themselves. So as a biospecimen provider, we see an opportunity for improved utilization of our clinical collections to help address some of those challenges. And biospecimens are really the foundation of translational research. Without diverse samples and, like I mentioned, that relevant data set associated with them, we can miss really critical biological signals that can affect disease detection and drug efficacy in these underrepresented populations. As we know, different populations can have distinct genetic risk factors. They can have differences in disease progression. They can also have comorbidities that may impact diagnosis and treatment. So these differences in gene expression, proteomics, and pharmacokinetics can be missed if you just have one homogeneous cohort and lead to results that are misleading and, like we said, maybe misdiagnosis in certain populations. So as we covered a bit, I don't want to repeat a little bit of what Tina said, but BioIVT offers these solutions to tackle some of the translational challenges that we're experiencing in the neuro space. It's all organized under our Center of Excellence umbrella, Center of Neuro Excellence umbrella. And I'm going to go into some details on exactly what that cohort looks like from a Alzheimer's and Parkinson's perspective, but really calling out that these are longitudinally collected samples. We have both normal healthy controls as well as disease cohorts across a number of different neuro-related disease indications. And really, it's an ideal resource for looking at disease progression studies with our longitudinal samples and healthy match controls to address those really complex research questions. So when we think about specimens, for many years it's the physical product, specimen tissue, blood sample that you're using for your research. But I think, again, as we're hearing more and more about AI and machine learning, it's more than just the specimen. Honestly, the specimen is useless without the data that comes with it, because then you don't know where it came from, what are the influencing factors, what's the biomarker data that's associated with it, all of that clinical history. The rich annotation is really the critical factor. So with those details in medical history, you can start to understand the characteristics of the specimen and, again, address those questions that you have. So you'll see here the extensive coverage that we have on the right side is our demographic details. We also, and you can see called out in there some of the lifestyle factors that we capture. We also have cognitive tests, which Tina mentioned, including MMSE and MoCA and others, as well as a number of commonly assayed biomarkers. And this is kind of like the framework that we provide, but again, we work and partner very closely with our clients because depending on the research question, there's additional annotation and things like that are needed, again, as you're digging into those complex research studies. And just to kind of go back again to the AI machine learning, there's a really strong need for well-curated data. You don't know how many times we have conversations with clients where maybe they're looking at samples that they've historically collected through clinical trials and elsewhere, and there's a lot of gaps. And I think that's one of the things to call out from a BioIVT perspective, just the well-curated nature of the data that we can provide is really making it analysis ready to feed into the systems that you're using to kind of probe those correlations and better differentiate patients, predict outcomes, and potentially assess treatment responses. So for our clinical collections, we can obtain up to 6 different sample types from each donor. And that allows, again, just a variety of different matrices depending on the research question that you're asking. And it also allows that comparison at the individual level. If you're looking at some comparison maybe between CSF and a blood-based biomarker, that opportunity exists in the sample collections that we have. So we have everything from, obviously, CSF. We have cell pellets from CSF and blood, serum. We have a variety of different collection types that are kind of enabled for liquid biopsy research, such as Streck tubes, ctDNA plasma. And all of that comes with that comprehensive HIPAA-compliant clinical data. And then as you can see here along the bottom, this is some information I pulled just on the demographic composition of the cohorts that we have. We have a really deep pool of patients representing both gender and ethnic and racial differences. And although it's not 100% correlated with the demographics of the disease, I think it's a great cohort, again, to probe, and I think there can be a lot of things teased out, which are sometimes lacking if you look at other biobanks that maybe have more of a European ancestry, or even within a U.S. population, a very skewed kind of racial or gender composition within them. So the takeaway is access to diverse samples is obtainable, and I encourage you to kind of explore what samples and cohorts that we have in our inventory. And just kind of paired with that, it's not just the diseased individual samples that we have. We also have those true normal. And so, you've heard of kind of normal specimens, but what is normal? I think again, the unique differentiators within our portfolio and what we call our true normal control specimens is that they've been validated to be normal. And the other kind of pairing to that is that they also span a diverse background, whether if you're looking at age, gender, as well as different ethnicities. And so these samples are from donors with normal cognition. They've been verified to have no history or family history of neurological or neuropsychiatric disease. They have no neurological complaints. And so it really ensures that you all as researchers are equipped with samples that are not just labeled normal, but truly embody that physiological normalcy. So the takeaway is that we have to begin to ensure that proper representation And, from our perspective, we see a lot of opportunity in the preclinical stages of research. Sometimes there's a challenge in considering that because there's a thought that, oh, if I have to go after all those samples and figure out where I'm going to source them from, that's going to impact my development timelines. But as you can see, these are all samples that are ready to go and, can immediately be built into research programs. So as a population diversifies organically, there's definitely a greater need, and I would stress urgency to make sure we're supporting that generalizability of findings. So with enhanced data across all of these demographically diverse cohorts, there's a more informed development of clinical guidelines. Also, from just a discovery perspective, if we think about genetic, proteomic, epigenetic profiles, those can all be influenced by race and gender. And so without looking at those in the early stages of discovery, we can potentially miss unique targets. Those can go unnoticed, undiscovered, and really have a huge influence on the potential opportunities for precision medicine. And finally, if we think about compiling representative data early on in the drug development or even diagnostic development process, we have to think too about down the line when we're thinking about clinical studies and having data that may help in that recruitment process. So again, if we can generate data showing, look, we looked at very, from patient samples of patients just like you that mirror your demographic and your kind of life experience, I think that will really help to build that trust and that confidence in the populations that we're trying to enroll in clinical trials and engage in the science that we're doing. So time is another critical factor. Biomarkers, as we know, can be dynamic. So longitudinal sampling really allows us to capture those disease trajectories. And again, just kind of tying it back to diversity, we know that these can change across racial and ethnic lines. So there's two types of study approaches, collection approaches. The longitudinal side, without kind of calling out all of these, but it's really important for teasing out those cause and effect relationships that can occur between different variables. And they're noted for their ability to monitor changes over time and establish that causality, offering ultimately more deeper insights. And that's the converse to cross-sectional, which is typically a more observational study. It really just kind of hones in on that snapshot of a population's current state, but it can struggle to track progress or identify those cause and effect relationships. So it comes down to essentially what question are you trying to answer and the type of study that you want to design. But I would call out, as it relates to biomarkers, we know that these are influenced by a variety of factors, and the impact of time really has to be considered as part of that. So again, from a perspective of BioIVT, we collect longitudinal samples across a variety of time points, and those are kind of matched to the disease indications. So with these studies, or with these samples, you can investigate things such as disease evolution and really dig in on the trajectory of that disease progression over time within individuals or across diverse groups. You can unpack some of those complex interactions, understanding how life course exposures, such as potentially chronic stress, can influence biological processes. And you can identify sensitive periods. So understanding maybe temporal relationships that become risk factors and can influence later development of pathology. These can be translated into potential prevention strategies. So integrating the longitudinal study designs with comprehensive biomarker measurements in that diverse cohort is really essential for understanding the multifaceted nature of AD and PD. And as we've previously covered, there's greater variability in minority cohorts. So just to kind of wrap quickly here, so we're seeing momentum. We're starting to hear regulatory groups and research foundations that are prioritizing diversity. And the industry is recognizing the commercial and scientific value of inclusive data. And I think that's really important. I think we're driving towards that scientific validity that's so important as we're thinking about precision medicine. So there's a number of opportunities out there, and I'm happy to see them kind of being moved forward. So just from emerging frameworks, there's examples out there such as the Women's Brain Project, and they're working to address some of those issues around sex and gender gaps in Alzheimer's disease. There's also the Alzheimer's Disease Neuroimaging Initiative, and they've actually put out their commitment to recruit 50 to 60% new participants from underrepresented populations. So exciting things to see there. We have technological advances, like I mentioned, blood-based biomarkers and AI-based modeling. And I think just, again, as we're considering that, it's important that from a digital perspective and as they're building out these foundational models to really make sure we're asking questions of what was the basis of the data set that generated that, and was it properly representing the patients that we're trying to treat, because it's a large volume of data, but if it's not representative, we're going to build in biases. The importance of community partnerships, like I mentioned, really engaging communities, patient advocacy groups, and that. And again, I think there's a lot of partnerships that we're seeing come forward. It's really exciting to see also these foundations partnering with other analytical groups really to dig in on some of the biomarker discoveries that are happening. And then finally, an industry shift. So making sure that as a collective research community that we're advocating for regulatory and funding priorities for more inclusive research. So ultimately in precision med, we're seeking to get that right drug to the right patient at the right time. And how that pairs to the points that I want to kind of drive home today is diversity in research. It's not just about representation, but through that improved scientific accuracy and equity, like I was mentioning, we're driving towards that scientific validity. It's also about observing differences in neurodegenerative diseases across different ethnic and racial groups and genders, and that's critical to make those more tailored interventions and precision medicines. And I think by looking at these diverse cohorts and considering really these very complex diseases, right, multifaceted biological processes, and all of the social factors that go into that may influence disease, we can accelerate further discoveries and medical breakthroughs. And then finally, the commitment to diversity. I think that goes a long way in helping to build trust in research and medicine. The more they see these things succeed, and again, succeed in the populations and communities that they're in, the more success we'll see in developing a more inclusive healthcare future. So with that, I think I'm right at time or a little over, but I wanted to thank you for your attention today. Happy to take any questions, or we can certainly talk during the coffee hour if we need to move ahead.